So to continue with the second thrust of my lab, which I would say is a more mature thrust of my lab, I started off in Purdue Computer Science on doing computational genomics, and my first grant was a National Institutes of Health grant on developing cyber infrastructure for metagenomics workloads. This was with Argonne National Lab, and they have this compute infrastructure for microbiome or metagenomics workloads, and it's called MGRAST. So on the computational genomics side, we have been working with both developing cyber infrastructure and also, if you think of the more data sciencey part of computational genomics, I'm very interested in the epigenome. Right? Now, the epigenome is the layer pervading the genome. It is essentially the chemical modifications on top of the genome. And it is multidimensional. There are many different types of modifications that you can look at in order to see, how dynamic the genome is or the epigenome is to changes in the environment. So some of these modifications are histone modifications, methylation, microRNA, etc. So we have looked at both histone modifications, which is a type of epigenomic modifications, and we have looked at great depth on these regulatory RNA, the area of regulatory RNA, and these are becoming very exciting these days because you have these RNA vaccines, mRNA vaccines that are there in circulation. So the first two vaccines that have been approved by the FDA in the United States are both RNA vaccines, and RNA is a very versatile molecule, right? So our initial work, where we also won a best paper award from ACM BCB, a top bioinformatics conference, is what we wanted to do is, when you have these regulatory RNA that are targeting different regions of the gene or messenger RNA, then you want to make sure that the targeting is precise. There is minimal off-targeting, and this can also be extrapolated to the area of genome editing. So if you think of CRISPR, the Nobel Prize winning, chemistry Nobel Prize winning discovery this year, right, with Jennifer Doudna's lab, you have these CRISPR, the CRISPR technology, where you have the single guide RNA, right, sgRNA, that is targeting a region of the genome or messenger RNA, and once it targets the right region, what it does, it goes and snips at that specific targeted area. So you want to make sure that the snip is happening at exactly the targeted area and there is minimal off-targeting, right? So our lab is very interested in increasing the precision of this SNP, right? So you want to make sure that the SNPs occur in those areas of the genome or those areas of the mRNA where they are intended to be. So our algorithms, we came up with our first algorithm called Avishkar. Avishkar is the Sanskrit for discovery, and our first algorithm was a simple SVM, support vector machine-based algorithm, and was a binary classification algorithm where we said, if you consider, say, a single guide RNA, we said whether this mRNA is indeed a target for the single guide RNA or not. So it's a binary classification problem where we used an SVM as a classifier. So you can think of this as an SVC, support vector machine classifier. But then we were interested in finding out if we could actually regress the strength of this interaction. So we used SVRs, so support vector machine based regressors, right? Where we could say that what is the intensity of this interaction, whether you're considering the interaction between a regulatory RNA and a gene, or whether you're considering the interaction between a single guide RNA and the area that is being targeted by the single guide RNA of this CRISPR-Cas9 technology. So a little bit about the CRISPR-Cas9 technology. Cas9 is the enzyme that is resulting in the SNP, right? And the single guide RNA is the RNA that is guiding this enzyme to the targeted region. So I think these, RNA technologies are very versatile technologies, and they're also being used for vaccines, right? So both the Pfizer vaccine and you have both of these vaccines that have been now released in the US. They are both RNA-based vaccines, right? So RNA has many different uses. The use that our lab has looked at is, can you decrease the off-targeting that occurs when you're using regulatory RNA microRNA, or when you're using CRISPR-Cas9 technology to target specific regions of a gene, right? So let's use genes synonymously with messenger RNA or mRNA. So we came up with Avishkar, where we had the ability to say whether a target region of a gene was indeed a target for a regulatory RNA or not. Then we had a distributed version of this algorithm where we wanted to make sure we can do this in an efficient manner. So we came up with a distributed version of this SVM support vector machine based classifier, and we have released this. This is open source, and this is based on, it has Apache Spark at the back end. And then what we then thought that, it should be in a context-driven manner. So for example, if you consider a healthy person and if you consider a person who has COVID-19, so there might be differences in the microRNA, mRNA interactions in a healthy person and in a diseased person, right? So can we do these interactions, both the binary classification and the regression, in a context-driven manner? That is, what is the physiological status of this human being, right, or of this organism? So that is again published, and we have both in theranostics, which we came up with the algorithm called Teresius, and then also in TCBB. So these were actually to show that if you have different kinds of cancer, then in different types of cancer you have different regulatory networks, and depending on the kinds of interactions that are taking place between the microRNA and the gene, we can do the mapping of these interactions in a context-specific manner. Right? So for this, we used Lasso for our TCBB paper. And Teresius, what we did is we actually had a very nice way to break down the problem. So what we did is we took two ANNs. ANNs are artificial neural networks. So we have two ANNs. And then the way we connect the two ANNs is using a Bayesian connection. So let me give you a little bit of a brief spiel on these two ANNs. So what the first ANN does is it tells you if mRNA or a gene is a target for a microRNA or not. This is what the first ANN is doing. And if it indeed is a target, then what the second ANN does is it regresses the interaction strength. So we have two ANNs. The first one is saying whether or not a gene is a target for a microRNA, and the second one is saying what is the strength of this interaction if indeed it is a 1. That is, the gene is a target for the microRNA. And then we connect the two ANNs using a Bayesian network in order to decrease the number of hyperparameters. And what this Bayesian connection does, it sees the sparsity of these interactions. So that's how we are connecting the two ANNs. So you can read all of our papers. We have a BMC genomics paper from 2015, where we started off with Avishkar, and then we had the ACM BCB Best Paper Award the following year, and then we have the TCBB paper and the Theranostics paper, where we got interested in seeing how different contexts, depending on the physiological context, these interactions between the microRNA and the genes can be different. Right? And again, this has implications in terms of CRISPR-Cas9 technology, because again, differences may occur because of the different physiological states, and depending on that, you might have sensitivity in terms of where exactly the single guide RNA will bind, right? And one of the important things in any of these interactions between these regulatory RNA or the Cas9 and the gene is that you want to make sure the chances of off-targeting is minimized, right? And this is one of the important areas or important applications of our technology, that decreasing the off-targeting, and by decreasing the off-targeting, you can actually take the technology out into the wild, right? Where you can be 100% sure, or very sure, or have a high probability of thinking that your intended interactions are what will be the majority of the interactions. And if there are indeed some interactions that are kind of the off-target interactions. They are the minuscule minority of the interactions, right? So I think the machine learning side, where you have epigenomics-based applications of our technology, and the data engineering side, where you have cyber infrastructure that enables the distributed forms of these algorithms, are both exciting computational genomics advancements in my lab, ICAN, that is the Innovatory for Cells and Neural Machines. So I hope to talk more about our advancements next year, but this is a summary of what we have done as of December 2020.